The dangers of erythritol, brain cell damage and stroke risk revealed. A popular sugar substitute found in many low-carb and sugar-free products may be damaging brain cells and increasing the risk of stroke, according to new research from the University of Colorado Boulder. The study, published in the Journal of Applied Physiology, sheds light on the potential downsides of erythritol, a sweetener that has been considered safe for decades. But here's where it gets controversial. Could this common ingredient, often touted for its negligible impact on insulin levels and low calorie count, actually be harming our health? The researchers treated human brain blood vessel cells with erythritol and observed significant changes, including reduced nitric oxide expression, increased endothelin-1 production, and diminished natural clot-busting compound TPA. These alterations suggest that erythritol consumption could lead to constricted blood vessels and a higher likelihood of stroke. While further studies in people are needed, the authors advise consumers to monitor their intake of non-nutrient sweeteners like erythritol. This study challenges the notion that sugar substitutes come without negative health consequences, sparking a debate on the safety of these widely used ingredients. What are your thoughts on the use of sugar substitutes like erythritol? Have you experienced any adverse effects? Let us know in the comments below. Scientists warn. Popular sugar substitute erythritol linked to brain cell damage and stroke risk. Imagine this. The sugar substitute you've been using to manage your weight or blood sugar levels might be doing more harm than good. Recent scientific findings have cast a shadow over erythritol, a popular sugar substitute often recommended for individuals with obesity or diabetes. This sweetener, found in a myriad of low-carb and sugar-free products, from ice creams to protein snacks and sodas, may not be as harmless as once thought. But here's where it gets controversial. New research from the University of Colorado Boulder suggests that erythritol could be linked to brain cell damage, potentially increasing the risk of stroke. Let's dive into the details. Erythritol, a sugar alcohol approved by the FDA in 2001, is derived from the fermentation of corn. It's a favorite among health-conscious consumers because it contains almost no calories, offers about 80% of the sweetness of regular sugar, and has minimal impact on blood sugar and insulin levels. These qualities make it an attractive option for those on weight loss journeys, low-carb diets, or managing blood sugar levels. However, recent studies have started to uncover some alarming risks. One study involving 4,000 participants in the US and Europe found that individuals with higher levels of erythritol in their blood were significantly more likely to experience a heart attack or stroke within the next 3 years. This finding prompted researchers at the University of Colorado Boulder to investigate further. Led by Christopher DeSouza, professor of integrative physiology and graduate student Auburn Berry, the team conducted a study published in the Journal of Applied Physiology. The researchers treated human cells that line blood vessels in the brain with erythritol at levels similar to those found in a typical sugar-free beverage. The results were striking. The treated cells showed significant changes. They produced less nitric oxide, a molecule that helps relax and widen blood vessels, and more endothelin-1, a protein that constricts blood vessels. Additionally, when exposed to a clot-forming compound called thrombin, the cells produced less of the natural clot-busting compound TPA. Furthermore, the cells generated more reactive oxygen species, ROS, also known as free radicals, which can damage cells and inflame tissue. So why does this matter? If your blood vessels are more constricted and your body's ability to break down blood clots is impaired, your risk of stroke increases. This is the part most people miss. The study used only a serving size worth of erythritol, so the impact could be even worse for those who consume multiple servings per day. It's important to note that this study was conducted in a laboratory setting using cells, and more extensive studies in humans are needed to confirm these findings. However, DeSouza advises consumers to be mindful of their erythritol intake. He suggests reading labels carefully and looking out for erythritol or sugar alcohol. Given the epidemiological study that inspired our work, and now our cellular findings, we believe it would be prudent for people to monitor their consumption of non-nutrient sweeteners such as this one, DeSouza said. So what do you think? Should we be more cautious about our use of erythritol, or do you believe more research is needed before we make any drastic changes? Share your thoughts in the comments below.